Hi everyone, my name is Gong. I'm a PhD student at Dr. Hong Chen's lab in Washington University in St. Louis. Today, I'm honored to present our work titled Comparison of Sonication Patterns and Microbubble Administration Strategies for Focused Ultrasound-Mediated Large Volume Drug Delivery. Diffuse intrinsic pontine glioma, or DIPG, is the deadliest brainstem tumor that spreads to the large area of the brainstem. It is very hard to treat, partially due to its often intact blood-brain barrier, or BBB, as illustrated by this T1-weighted post-contrast enhanced MRI. In recent years, our group, as well as others, have shown that it is feasible to open the BBB at the brainstem in DIPG models with focused ultrasound or FUS sonication. For clinical translation of this promising technique, efficient and homogeneous large volume BBB opening at the brainstem is critically needed to treat DIPG with therapeutic benefits. In the recent decade, large volume focused ultrasound BBB opening strategies have been developed and applied in many preclinical and clinical studies. It is usually achieved by sonicating multiple points within a defined treatment grid, and the pattern is either interleaved when the sonication is iterative, or serial when each point is sonicated separately without looping. As another important factor of BB opening, microbubbles are often injected either through bolus injection or continuous infusion. Given all these options from this list, an unanswered yet important question is that which one should we choose to achieve efficient and spatially homogeneous large volume drug delivery? What is the optimal strategy and why? So the objective of this study is to determine the optimal strategy to achieve efficient and homogeneous large volume BB opening at the brainstem. We identified the sonication pattern and microbubble administration method to be the two critical features. So we compared the four combinations and determined the optimal strategy, and then applied it to deliver an immune checkpoint inhibitor, anti-PD-L1 antibody with fluorescence label. An ultrasound image-guided focused ultrasound system was used to perform the treatment. A single-element FUS transducer was mounted to a programmable 3D positioning stage that moved within a 3x3 treatment grid at the mouse brainstem. An ultrasound imaging probe was co-aligned with the FUS transducer at the center opening to provide both targeting guidance as well as detecting acoustic emission from microbubble cavitation. A catheter was placed at the tail vein for an intravenous injection of microbubbles, Evans-FUS model drug, and PD-L1 antibody. The first sonication lasted for 3 minutes, and the mouse was sacrificed 40 minutes after the treatment. The brain was harvested and quantified using ex vivo fluorescence imaging. Here shows the brain slices for 4 combinations with bright field images on the left and the corresponding fluorescence images on the right. All cases showed successful large volume BB opening, illustrated by Evans-FUS extravasation at the brainstem. We quantified the efficiency using total fluorescence intensity at the brainstem, and we found that interleaved sonication combined with bolus injection yielded the highest delivery efficiency among the 4. We also quantified the cavitation dose values at all points within the treatment grid and obtained a consistent trend for total cavitation dose. From both quantification results, we found that bolus injection of microbubbles led to a higher fluorescence intensity and cavitation dose than continuous infusion. We then quantify the spatial homogeneity of BB opening using coefficient of variation. A lower coefficient of variation implies higher homogeneity. From the plots, we can tell that interleaved sonication can lead to more homogeneous delivery than serial sonication. Therefore, to combine the two findings, we pick interleaved sonication with bolus injection to be our optimal strategy among the 4. So using this identified strategy, we then deliver the fluorescence label PD-L1 antibody using 3 different pressures. And we showed successful delivery under both 0.3 and 0.45 megapascal, but not 0.15 megapascal. The corresponding cavitation dose was also quantified, and we found a strong correlation between delivery efficiency and total cavitation dose with R squared equal to 0.81. This implies that cavitation can be used to monitor the large volume BB opening outcome. Last but not least, we evaluated how safe it is to have large volume BB opening at the brainstem. From safety staining, we found no signs of hemorrhage at the two low pressures, but we found some microhemorrhages with 0.45 megapascal. The hemorrhage density with respect to the whole brainstem was quantified. This shows that lower pressure can decrease the risk of causing microhemorrhage by large volume sonication. So to conclude, this study shows that efficient and homogeneous drug delivery by large volume FUS-BBBO can be achieved using interleaved sonication combined with bolus injection of microbubbles. Second, cavitation can monitor the efficiency and homogeneity of large volume FUS-BBBO opening. And third, lower pressure such as 0.3 megapascal is preferred to achieve both efficiency and safety for large volume drug delivery at the brainstem. As for our future work, we will further evaluate the therapeutic outcome of large volume drug delivery in a DIPG model. I would like to thank Dr. Hong Chen and all my lab mates for their help and support on this project, and here lists all the fundings. Thanks for your attention, and I'm happy to take questions. Thank you.